And good afternoon, microbiology friends. So standing like this, I can see my hair is really getting thin. But we're going to get into Chapter 7, which is control of microbial growth. We're going to be talking about some of the technologies and principles behind sanitization, sterilization, things like that. Actually, some things that are quite relevant with all the controls being used, the quote, deep cleaning and hand sanitizers, how they're actually working. We'll be getting into. This is not so much conceptual as it is kind of lists of things and ideas and some vocabulary. So I thought that we could use the PowerPoint fairly effectively. I'll post the PowerPoint as well. And let's see. So exams, I'll be honest, I haven't been on top of grading them. I've been having to take care of other classes and things. But we'll get on them this weekend. And as far as the article stuff, I haven't figured that out yet, but I'm working on it. It was an article review that we would do as part of an exam kind of boost. Hopefully everybody did okay. But I'll get into that as we move forward next week. And yeah, so control of microbial growth. Why do we need to control it? Well, actually some interesting things to think about with this because, for the most part, here I'm going to crank you up a little bit higher, there you go. For the most part, we don't really want to control microbial growth. That, the excessive use of the excessive use of antibiotics leads to resistant species. The excessive use, you may have heard, of really harsh soaps isn't good for the microbiome on your skin. Bacteria do all kinds of really wonderful things, and sterilizing the soil is a great way to keep plants from growing. And so, controlling bacteria isn't necessarily something that you always want to do. It's important to have a good crop of bacteria. I like to think of our bodies as being one great big bacteria parking lot, and you want to keep it filled with the non-pathogenic bacteria, bacteria that are good to have on you, and that way you don't leave space for bacteria that you don't want to have on you. That's why sometimes when people take antibiotics, they can end up with a yeast infection, different things like this. So we don't necessarily need to control bacteria all the time, but there are a few cases, obviously, with a bacterial infection that we really want to. Food is another place where you want to control it. We'll get into this. So just some things to think about, that things that bacteria are doing for us. They're kind of outside the notion of medical microbiology, but still, bacteria are critical for fixing nitrogen in the soil. So sterile soil without a source of nitrogen isn't going to be very good to grow plants in. Inside our bodies, bacteria are producing vitamin K and a variety of other different kinds of molecules. They are digesting food both inside cattle and inside us. They are very important for being able to break things down and put it back into the environment for other organisms to live on. Bacteria can be used for this idea of bioremediation, that is, putting bacteria in a oil spill. There's even work now, bacteria that can break down plastic. So bacteria, they're our friends, and I agree. If they don't bother us, let's not bother them. That said, there are some very good reasons to control bacteria. Obviously, for our purposes of medical practice and stuff, bacteria that cause disease, that is not a good thing. Passing the disease-causing bacteria to others is not a good thing. You may not have it, but you may have it on your hands and you're preparing food. Don't even get into that. Things like pollution control systems can get thrown off if bacterial growth is allowed to run rampant. Algal blooms, while not strictly prokaryotes, but a lot of microorganisms, including bacteria, they can really get going in lakes and even in the ocean. And one of the interesting things about this is they can begin growing very quickly, they can then begin pulling nutrients out that maybe the fish versus the other organisms would need, but they are consuming the oxygen. And then when they die off, the oxygen is incredibly consumed as this material is beginning to break apart, and it generates essentially an anaerobic environment in a lake, and this is known as eutrophication. It is a buildup of toxins and materials in lakes. So sewage treatment plants actually have to worry about this. When they are testing sewage, the effectiveness of the actual sewage processing is carried out by looking at the amount of material that's in the water that would allow bacteria to chew on it and reduce the oxygen. It's called the biochemical oxygen demand, or the BOD. And so if you go to a sewage treatment plant, that's one of the things they're checking every day, is the level of organic material in the water, the effluent, and the ability of bacteria to chew on that and suck the oxygen out of the water is a bad thing. And so what you want to do is have the sewage treatment in the plant work so lots of bacteria are growing on it and eating it all up, and lots of oxygen is being introduced into it. So then when you dump it out into the Sangamon River, you don't have this biochemical oxygen demand anymore. It's already been consumed. Spoilage of food is another very particular issue. That bacteria getting into food cause it to be unhealthy. They can begin to destroy the food. And if you want to increase the shelf life, you got to control the bacteria. And so, things like canning, that's what you're doing, is you're controlling bacteria growth. So, what we can do is think about some of the techniques that we use to kill bacteria. And we can break this down in a couple of different ways. We're going to kill or inhibit the growth of nasty bacteria, and we try to do this without harming the rest. And so some of the things that we do are going to be actually killing everybody, but other things we're doing are stopping anaerobic bacteria from growing by providing plenty of oxygen, or blocking oxygen in other cases. So just as a little list here, things that you probably, if I were to ask you, what are some of the things that we do to, for general cleanliness, general bacterial cleanliness? You wipe down tables, you kill bacteria in the food with heat or with osmosis, adding lots of salt, or removing the water, drying things. That works pretty well. You can block oxygen from entering the soil, and that can stop the acid production of the bacteria that are there. They will slow up their growth and not dump acid. Putting things into the refrigerator, low temperature, slows down bacterial growth. We can use antibiotics. It's a really obvious thing to do. Blocking the air or water flow in different environmental areas can do this. And so, in a general sense, we can break the different kinds of growth control systems into either a physical or a chemical means. And we're going to break these apart and look at them a little bit, okay? So here are some of the things to think about in terms of some vocabulary and sort of, a list of goals that we might want to think about for reducing bacterial load. So we had these two very similar terms: sterilization and sanitation. Now the difference is sterilization is we have absolutely killed all the life in the solution or in this on this material. Sterilization is pretty darn hard, but we can sterilize things by putting them in an autoclave, a high pressure cooker, baking them at 300 or 400 degrees. That will do it. There's different kinds of filtration systems that'll do this. Something that's sterilized, though, you probably have to keep it packaged up or keep it in a sterile environment to maintain its sterility. And for some things, it's not all that practical. Now, sanitization is reducing the numbers of bacteria to a level that isn't going to make anybody sick, and because you've reduced the level down, it takes a while for the level to come back up, and you have now prolonged the item. A perfect example of sanitation might be things like pasteurization of milk. You may or may not have experienced milk going sour in the refrigerator. Well, that's because, even in an unopened container, the pasteurization process is not a sterilization process, because sterilizing the milk is not all that easy to do. You end up having to heat it. Milk doesn't respond very well to heat. You end up with something that tastes a little bit off. You have canned milk, but if you've ever tried it, canned milk doesn't taste nearly the same as fresh milk. And I've never had non-pasteurized milk, but regardless, pasteurization is going to drop the load way down, but not completely inhibit it. So even though you haven't even opened a carton, you put it in the refrigerator, and you let it go there for a couple of months, open that puppy up, go ahead and get your roommate to try it first, because it's going to probably have gone off even faster if you take an unopened carton of milk, and rather than putting it in the refrigerator, let it sit on your counter for 2 weeks, see how that goes. So there are ways that you can sanitize, or that you can actually sterilize milk using things like radiation. You can find milk now in boxes that is pretty much room temperature stable for a very long time. Those actually have been treated with different techniques rather than just the standard pasteurization, which is a brief heat treatment to knock the level down, not to zero. So sterilization is hard. Sanitation is not all that hard to do. One of the things that we maintain is something that is known as aseptic technique, and that is different kinds of strategies that will minimize or inhibit the transfer of bacteria from one source to another. So everything from wearing gloves, not coughing on stuff. Aseptic technique is practiced all the time. You have a hypodermic needle, you keep the cap on it as long as possible, you wipe the skin down with alcohol, you maybe even wipe the needle with alcohol, and these are things that are maintaining a low level of bacteria as much as possible. There are some things that, if you sterilize something and then you open it up into the atmosphere, it is no longer sterile. Within seconds, there's going to be bacteria that are probably falling around on it, and certainly if you touch it. So you have to be very careful in maintaining aseptic technique. Disinfection is a way that we can use fairly nasty chemicals that kill bacteria. The problem with disinfection is, in order to kill the cells of the bacteria, you're using stuff that may very well kill your cells. So our skin is pretty tough, and there is a range of disinfectants that we can use on our skin that you'd never want to take orally or inject. Yeah, don't be doing that with bleach. Don't even wash your hands with bleach. But antisepsis this is sort of the difference between sterilization and sanitation. Antisepsis is really now removing bacterial load, but not down to zero, not as good as disinfection, and we're using things that are less toxic. You can use things that are on your skin. So if you've got a glassware or surgical equipment that you need to disinfect, there's all kinds of things that you can do to that you'd never want to do to your body. All right. There are a range of different kinds of chemicals. We've got chemicals that will either kill the bugs, and these are two kind of important terms. That is known as something that is bactericidal, or we have chemicals that will inhibit the growth of the bugs, and this is referred to as being bacteriostatic. So it didn't kill them, but it just made it so they can't replicate right now. And there's a little bit of a difference with that. Antibiotics are using these two different strategies. Some antibiotics are bactericidal. They're killing bugs dead. Other antibiotics are inhibiting the growth of the bacteria as long as they're in the presence of that antibiotic. We'll look at some of these in just a moment. So one of the things that's interesting about this is that we are taking advantage of the fact that prokaryotes are really a good bit different than us as eukaryotes. There are things that bacteria do that we don't do, like having cell walls, and there are subtle differences in the internal structures, the material that makes things like proteins or makes RNA polymerase in the ribosomes, that are just different enough from ours that we can develop drugs that will block like protein synthesis or RNA synthesis in bacteria but not affect a eukaryote. So one of the things that's interesting to think about is if you get a eukaryotic cell infection, like a yeast infection or some kind of eukaryotic parasite, those are often a lot harder to kill because you're trying to find something that will kill that eukaryote and not this eukaryote. So bacteria, they've got enough differences that it's not too hard to find targets that are different. But other eukaryotes, it's not all that easy. All right. So a couple of things that we think about that make a bacteria control system useful or not so useful. You got to be careful if you're going to be using it around people. It can't harm people. All right, radiation, not a good thing. I mean, we can absolutely irradiate a room with either, God forbid, gamma irradiation, radioactivity, and man, that'll kill everything, sterilize it. We can, maybe you've seen at the hospital, these little robot machines that kick out a huge amount of UV light. UV light is very ionizing. It splits water to create free radicals. It breaks DNA up. It is not good to be exposed to excessive UV light. I mean, you can go from getting a suntan to getting a sunburn to getting skin cancer really, really fast. So there are things that, and conditions that you just can't use certain control systems. Other things, you don't want to damage the sample necessarily. Things like glassware are pretty darn tough. Metal, even tougher. So there's a lot of stuff you can do with that. But stuff like food, well, you can't heat necessarily all food up to a temperature that'll kill all the bacteria to sterilize it and still maintain its integrity. I mean, there's some foods that just don't respond very well to that kind of heat. So that's another thing that you have to think about. Another thing that is thought about is the expense. You probably can make sterile milk, but it does cost a good bit, and it becomes cost prohibitive. Then lastly, things that are really complicated or that are dangerous to apply, probably not the best thing there either. If it's really complicated, it's easy to screw it up. And one thing you don't want to do is go through a procedure to sterilize something, skip a part or not carry out some aspect of it completely, and then end up with one of your instruments not being sterile because the protocol was very complicated. Not a great sterilization method there. So let's just go through some of the mechanisms by which the different sterilizing or sanitation techniques might work. So a classic way of sterilizing things is to heat it up. So one of the things I think about is the same principles that we use in cooking are the same principles that we think about for sterilization. Because really when you're cooking stuff, you're changing the flavors up. Yes, you're changing the textures up. But one of the main things you're doing is preserving it. And so for canning and things. But we can think of a couple of different ways to think of that. Temperature, though, is a great way, pretty much a great leveler. You bring anybody up to boiling, you pretty much are going to kill everything. There is not much on the planet that can survive being in boiling water for 30 minutes. That's going to get everybody. And what's happening? All those weak-ass bonds that are holding proteins together, they can't handle, particularly the hydrogen bonds, they can't handle boiling. The proteins denature. Without a functional structural protein, the enzymes won't work, no metabolism. Now, interestingly, something can be sterile and still not be very sanitary, and in fact still be toxic. If you've got a really big bacteria load, you can kill all the bacteria, but you don't probably want to eat that because it's not going to be A, very tasty, and B, there's all kinds of toxins that are floating around in it. So, take that for whatever it's worth, is that it is sterile, but it doesn't necessarily mean that it's clean. But if it's sterile, it's a lot easier to clean it than if it's not sterile. So by heating things up, we denature the proteins. Another great way to denature proteins is to add detergent. So heat and detergent is doing a great job, dishwasher. So not only are you denaturing the proteins, the soaps and detergents are breaking apart the membrane, forming micelles and things. And so that's a really good thing to do. So wash your hands in hot water with soap. Man, that'll do it. Although in that case, how about we wash our dishes in soap in real hot water using a dishwasher? That I like better. So, all right, with temperature, we got a couple of things. Part of it is that some bacteria can sporulate, and when they form spores, those are somewhat heat resistant. You have to really heat them for a while and a lot of heat. So we've got a couple of different ways of administering heat that is practical. One is an autoclave. This is basically a pressure cooker. And so you remember from chemistry, PV equals nRT. Maybe you don't remember that. The point being, you can raise the boiling point of water well above 212 degrees Fahrenheit or 100 degrees centigrade by increasing the pressure. That's what a pressure cooker does. So you can raise the temperature up well above the standard boiling point of water, really hot, by putting it into a pressure cooker and exposing it to steam. Two different ways of doing it. Anyway, this is going to kill pretty much everything because that temperature, that's going to do it. It can melt plastics. It can damage, it's going to cook food, and sometimes it makes it so it's not very palatable. It will potentially damage sensitive instruments. There are definitely a lot of machines that you can't do this. I mean, things with electronics, my God, no way. Boiling is not bad. Boiling at 100 degrees for a minimum of 10 to 20 minutes, and past an hour, you probably are just wasting your time and your energy. But this is going to kill all living cells. There are some endospores that may be boiling resistant. Just an example, if you ever go out camping and you have questionable water, certainly back in the day, one of the things you would do is you take the water and you boil it for 15, 20 minutes. And if you don't have a lot of good sanitation, but you do have heat, that's a really good way to sterilize or to make your water drinkable. So you can imagine, though, yucky water, you boil it, it's still maybe not all that palatable, but at least it doesn't have any living material in it. So that's our heat aspect. A couple of other things that are, you've heard of, the idea of pasteurization, which is a heating not at boiling, but a brief and cooler heating, above body temperature, something that's going to kill mesophiles, but not so hot that it's going to denature proteins, and you can then end up dramatically reducing the bacterial load. So it doesn't sterilize the food, but it does reduce the load, so it will have a better shelf life. An example here, you have fresh milk, you pass it through a heat exchanger thing where you've got some kind of heating coils or tubes. You can bring the milk up to a couple of different temperatures for different times. Things that are ultra pasteurized are brought up pretty high for a very brief moment, and then they are immediately cooled, and then that is going to dramatically reduce the bacterial load. Probably not going to kill everything, but it will dramatically reduce it. Other things you can do is to take materials and put it into an incinerator. These are things that we use in the lab, where you have a temperature that is, hotter than your oven, like a hot pizza oven or hotter than that. And that's going to essentially convert everything to either carbon or carbon dioxide. You're burning it all off. It's going to destroy a lot of things. If it's not metal, you can't really put it into an incinerator. But incineration, that'll do it. The other side of this coin is to bring the temperature down. So refrigeration is another technique. Now, refrigeration is probably not going to kill that many bacteria. You're going to make the bacteria's membrane a bit more rigid, and so they may not be able to replicate very well. There are bacteria that are called psychotrophs. These are bacteria that actually live in the Arctic or in ice and things, because you can find bacteria that live that way. They can actually operate, but they're probably not all that common. The main thing that refrigeration is doing is it's pulling the energy out that is needed for activation energy. So even though you have enzymes, if you don't have enough energy in the system, temperature, you can't get over that speed bump to carry out a biochemical reaction. So refrigeration does a pretty good job of that. Freezing is doing a really good job as well. Freezing, pretty much, your commercial or your home refrigerator is around 0 degrees Fahrenheit, or what in the centigrade world, minus 20. And commercial freezers can actually be colder than that. They might even be minus 40. I've got a freezer here in the lab that is at minus 70. You ever go by places that sell Dippin' Dots? In order to keep the little dots from sticking, you have to have an ultra cold freezer. It's not minus 70, but it's colder than a standard refrigerator freezer. So freezing is going to solidify the water, causes the water to crystallize, which often ruptures a lot of cells. It can denature proteins and things. You do end up with some dehydration and oxidation. So things can get what's called freezer burn. But freezing is a pretty solid way of blocking things from growing. So while refrigeration, not perfect, stuff will grow. You have the experience of stuff spoiling in the refrigerator. You put stuff in the freezer, it ain't going to spoil. I mean, you'll have other problems. It'll dry out, but it won't, in fact, support bacterial life. So let's see, a couple of other physical means that we can think about is the idea of filtration. And this is another common camping tool in which you can have a filter that will sterilize the water. And what you're doing is you're taking the water, pumping it through a filter, and you probably have to apply some pressure or vacuum on the other end, and you're pushing the water through a membrane filter that's got pores that are small enough to block bacteria, but large enough to pass fluid through. Now, to stop viruses, you have to have a really, really fine filter. It's going to take a ton of pressure or a really hard vacuum, and they're pretty expensive to do, but we can stop a lot of things. This works. So it is a very gentle process, so you can have a very complex bacteria media that you can sterile filter. And it's just something to think about. There are times where you have media that will have certain compounds in it that can't be autoclaved. If that's the case, you have to sterile filter them. So sterile filtering is not something you can use with solids. I mean, I would say duh, but maybe not that obvious. If it's also really viscous, like honey, you ain't going to be able to sterile filter that because you can't get it to go through. It's not great at removing viruses. Viruses are so small that to get a filter that is the size that can separate out viruses is going to require a lot of pressure. But filtration works really well for delicate, valuable, expensive fluids. You can sterile filter them. So then, let's see, a couple other filtration systems. We have a membrane, or you could have some kind of ceramic filter. And the filtrate is the term for what comes out. For air, or for gases, we can use a HEPA filter. You may have heard of these, a high-efficiency particle air filter. HEPA filters are able to do a pretty good job of removing dust and pollen and hair and bacteria. Again, not so good with viruses, which is an interesting thing, not to be overly picky or critical. We've got these HEPA filters now in multiple classrooms that are running and making a big roaring sound. Whether they're pulling viruses out, probably not so good. There are things you could do with air filters. If you have a UV light on them, that might work. If you have the filters that can generate ozone, that might work. But just putting the air through the filter, I'm not so sure how well that works. I thank the university for being thoughtful, but how well is it working? I don't know. I mean, why not try it? But it's not it's not a guarantee anyway. So, if you ever worn an N95 mask, these are very tight fitting. They've got very fine filters, and they are not all that comfortable to wear. It takes a little bit to breathe through them. Those can begin to block viruses. Viruses are pretty darn small. I mean, gases are obviously a lot smaller, but it's not an easy thing to do. It requires a fair amount of pressure to pull them through. So let's see. A couple of other, what we would call physical means. These are just various energetic systems, heat being one. Radiation is a great way to sterilize stuff. I mean, it will kill bugs dead. Radiation, we use the term ionizing radiation, which is essentially what particles that are generating radioactive decay are different kinds of isotopes and things will give off ionizing radiation. Specifically, we're talking about ultraviolet light, X-rays, gamma rays. These guys are very, very strong, powerful electromagnetic waves. And they're doing a couple of different things. They're either breaking apart DNA directly, shattering your DNA, and so you can't replicate anymore, or they are causing water to split to generate oxygen radicals. And oxygen radicals can then act as a really good bleaching agent. And so that's a pretty serious way. And there are other aspects of affecting the DNA that, depending on the wavelength of UV light you're doing, you can create what are called thymine dimers. It is essentially a coupling of nucleotides that blocks the DNA synthesis and it actually changes the nucleotide sequence. It can be very mutagenic, and that's going to cause the organisms to die. So X-rays are a brilliant way of generating mutations, and if you have very gentle X-rays, you can produce small DNA effects. If you've got blasting X-rays, you will create sufficient mutation and shattering of the chromosomes that the organism can't divide anymore. That counts for you as well. So this is one of those things that you probably wouldn't want to put people into. No, let's not expose people to UV, X-ray, gamma ray. Don't do that. But instruments, rooms, that works. Nobody ever uses things like gamma ray irradiation or X-ray irradiation. But as a tool for sanitizing food or sterilizing food, food irradiation is a thing that, whether it's actually still going on or not, the labeling of the food that had been irradiated was the radiation trefoil, that little three-pronged star symbol. And nobody likes to see their lunch meat with a radiation symbol on it. So then either the world has changed the labeling laws or they're no longer doing food irradiation. But food irradiation definitely works. Just as a little note, all the food that goes to the International Space Station has been irradiated, which means it is completely sterile. And it really is a perfectly harmless way of sterilizing food. The food itself in no way is radioactive. It's not activated anyway. You may have a loss of some of the vitamins. There's oxidation that's going on because you are producing these free radicals, but it's probably not that much more of a change of the food than other cooking mechanisms might be. So it's not fresh food, but or not as good as fresh food. Let's see. So a couple of things to think about is that these kinds of rays and energy are only going to be killing living cells. They're not going to be doing anything to samples, to equipment, stuff like that. The UV light doesn't penetrate very far, whereas gamma will. Gamma will go through stuff. UV light is pretty much just for surfaces. But like I know at the hospital, they will cordon off a room, put a little do not enter sign, and bring in the UV robot that is just shooting UV light all over the place, and that will do a pretty good job of sterilizing the room. So here's some examples of this. You've got a UV surface irradiation to generate a sterile work surface. Maybe if you've done any clinical work in a hospital, in the lab, they might have a sterile hood. And this sterile hood that we're seeing up here is actually probably a two-prong guy, in that it's got sterile air being generated where you pull air in from the bottom and you blow air out towards the sill or the sash. And it's what's called a laminar flow hood. The air is being passed through a HEPA filter, which is going to block and stop any bacteria and other microorganisms from passing through. And as long as you keep your hands in the on this side of the airflow, not actually behind your sample where your hands are stuff is blowing over your hands and onto your sample, you can actually work and keep things sterile. So we have a couple of sterile hoods here in the building. It is a way of doing aseptic work pretty easily. One of the things that you often do is you'll flip the switch on the sterile hood to begin blowing laminar flow HEPA-treated filtered air into the cabinet. Then you'll also turn the switch on for the UV light, and you have the sterilizing UV light. And so before you go into work, or to work at the hood, you'll hit the UV light for 30 or 40 minutes and let the fan blow, and that's pretty much going to kill everything that's in the hood. So there's no living material there. Then you have a whole room sanitation. Here we have a surgical theater that a great big, looks a little bit like one of the robots from Doctor Who. Great big UV light system is just blasting out huge amounts of UV light. Gamma ray irradiation is a process that is not used for rooms, but it is used for food, in that you have a gamma source that is kicking out gamma radiation and interacting with the DNA and things. So one thing is, here's the meat before and after treatment. It doesn't look that much different. You're not going to be able to clone the cow from this, though, because the cells are all dead. It's basically cooked, in a sense. At least in terms of the DNA, it's all shattered. And it is going to absolutely block any further bacterial growth. And one of the cool things you can do with this kind of stuff, because gamma rays do penetrate really well, you can package the food up, like put the milk in a box, put the bacon in a container, pass it through the gamma radiation. The package is sealed. Ain't nothing going to grow in there because it's all dead. You're done. So rather than having the fluid come out of the pasteurization machine and then pumping it into a bottle, you never even have to repackage this stuff. So another way of thinking about reducing bacterial load is actually taking on osmosis. That is, we can pull water out of cells. Remember, we talked a little bit about osmosis. Water follows salt or sugar. You can pull the water out by exposing the material to a real high salt or sugar concentration. And this is going to pull the water out like dehydration as a good food preservative technique. The reason preserved food works is that all the water's been pulled out, and so all these salts and things that were in that cell are now highly concentrated and stuff can't live in it. So you could dry stuff, you can salt things, you can put sugar in to preserve jellies and jams. So these are other kinds of bactericidal mechanisms. Now again, in terms of the health biz, probably not all that applicable, but there you go. We're going to stop here, and on Monday we'll get into some of the chemical types. So go ahead and have a swig of hand sanitizer on me. No, don't do that. Well, sure, go ahead. And we'll be in touch on Monday. I'll talk to you. All right, thanks for your attention. I'm Dr. G. Peace.